In this video, we are going to look at the neuromuscular junction and how the nervous system causes contraction in the muscles. So a motor plan, or the thought of moving muscles, starts in our frontal lobe in the motor cortex. And then a signal moves down the spinal cord to a motor neuron. And then the motor neuron is going to signal the muscles and cause an action potential in the muscles. And then that action potential causes the muscles to contract. So in this video, we are going to look at each of the steps from the motor neuron signaling the muscle to the process of muscle contraction. Motor neurons are the largest diameter neurons in the entire body, and they are very long. They go from the spinal cord out to the muscle that they are innervating. So they are myelinated. So the action potential can travel very quickly along motor neurons. This is just a reminder of where we left off in the last video. This is a bone. Our muscles are connected to the bone by tendon, and muscles are composed of bundles. These bundles are called fascicles. Within the bundles of fascicles are bundles of muscle cells. So we are focusing on this aspect today. We're going to look at what is inside the muscle cell. These myofibrils are bundles of proteins that are going to do the contracting. If we zoom into the muscle cell, muscle cells are surrounded by blood vessels. Skeletal muscle cells have multiple nuclei. The covering surrounding the muscle cell is called the endomysium. Underneath that is the membrane. The plasma membrane of a muscle cell is called the sarcolemma. And the liquid component, the cytoplasm inside, is called the sarcoplasm. Now these bundles are called myofibrils, and they are made up of filaments. There are thick and thin filaments, and we are going to focus on that in a few minutes. The other thing that I want to point out is inside the muscle cell, we have the smooth endoplasmic reticulum, but we call it the sarcoplasmic reticulum. That is all these blue tubules, these dark blue tubules. And they are important because they contain a lot of calcium, and this is extremely important for muscle contraction. The other thing I want to point out is this light blue tube here. That is called a transverse tubule. There are transverse tubules all throughout the muscle cell, and they actually open up to the surface of the plasma membrane. And this is an invagination of the plasma membrane. So the T-tubule is where the action potential is going to spread so that it can spread throughout the muscle cell. Now the T-tubule has to be aligned next to the sarcoplasmic reticulum because it's the muscle action potential that will trigger the release of the calcium, and we need this to happen for muscle contraction to occur. So, how do we get an action potential to occur in the muscle cell? The action potential has to be triggered by the nervous system. So here we have a motor neuron, and this is bringing an action potential down the motor neuron. So remember, when we have an action potential, we are going to depolarize the membrane and then repolarize the membrane. When depolarization is occurring, sodium is moving in. When repolarization is occurring, potassium is moving out. It's the charge change in the cell that is moving along the axon to the axon terminals. So this is showing our motor neurons synapsing with a muscle cell. I want to point out that one motor neuron can innervate many muscle fibers, up to 3,000 muscle fibers actually in a large motor unit. So a motor unit is when you have one motor neuron and those axon terminals are synapsing with multiple muscle fibers. So a large motor unit can have 3,000 muscle fibers being stimulated by that one motor neuron. A small motor unit would have fewer muscle fibers stimulated by that one motor neuron. So when that motor neuron synapses onto the muscle fiber, that connection point on the muscle membrane, that is called the motor end plate. And the end bulb of the axon terminal, where it connects with the motor end plate, that is the neuromuscular junction. So now we're going to zoom into what is happening in there. So we have our axons. This is only showing one single muscle fiber, but remember, many muscle fibers can be innervated by one motor neuron. The very, very end portion of the axon terminal is called the synaptic end bulb, and this is where the neurotransmitters are stored. Now, what is the neurotransmitter that is always released from motor neurons? The neurotransmitter that is stored in these vesicles in the synaptic end bulb that causes muscle contraction is always acetylcholine. And sometimes we use ACH as a short form for acetylcholine. So where the synaptic end bulb connects with the muscle cell membrane, this component of the muscle cell membrane is called the motor end plate. So let's zoom in a little bit more. Now we are just focusing on one synaptic end bulb. This is the axon terminal where the action potential is moving along. So that means sodium ions are moving in and changing the charge inside this axon to be positive. When the positive charge reaches calcium channels, these calcium channels are going to open. This is different calcium from the sarcoplasmic reticulum that I talked about a second ago. So calcium is going to move into the cell when there is an action potential in the motor neuron. And the calcium moving in is going to cause these vesicles containing acetylcholine to release the acetylcholine into the synaptic cleft. So the part of the muscle membrane where the acetylcholine is going to bind, that is the motor end plate. So these calcium channels, they are voltage gated because they open because of a charge change, which is caused by the action potential. Now let's focus in more specifically on what is happening on this membrane. So the action potential in the motor neuron causes calcium to move in, and this causes neurotransmitters to move out. They move out through a process called exocytosis, and they're going to go into this synaptic cleft. When the acetylcholine crosses the synapse, it's going to bind to ion channels. Now, these ion channels are chemically gated or ligand gated because they are going to open and let ions in because of a chemical, and the chemical is acetylcholine. So when these ion channels open, they're going to allow sodium to move into the muscle cell. This pink part here is the muscle cell. So sodium is moving in to the muscle cell. This dip in the membrane is showing the T-tubule. This is an extension of the muscle cell membrane that is dipping into the muscle cell. So number 1, acetylcholine is released. Number 2, acetylcholine is going to bind to chemically gated channels. 3, that is going to cause an action potential in the muscles. Sodium is going to move in, just like the action potential that we had in our nervous system. Sodium is going to move in for the depolarization phase, and potassium is going to move out for the repolarization phase. The action potential, how the mechanism works across the membrane, is exactly the same in the neuron as it is in the muscle cell. So muscle cells are excitatory like neurons. So the sodium moves in and it depolarizes the membrane. The only difference now is that the action potential is going to travel through T-tubules, the transverse tubules. So now the muscle membrane isn't just surrounding the muscle fibers, it's moving into the muscle cell, because that action potential, that charge change, has to move deep into the muscle cell so that it can trigger the calcium to be released from the sarcoplasmic reticulum. That calcium inside the muscle cell is very important. We'll talk about that in a second. So the action potential in the muscle is still moving across the membrane, just like the neuron, but it's moving into the cell through the T-tubules. And then lastly, after we start to depolarize this muscle cell, then the last step is that the acetylcholine is going to be broken down. I want to just focus for 1 second on this aspect. Previously, we talked about serotonin, how serotonin moves across a synapse and it binds to the postsynaptic cell. Serotonin is the neurotransmitter that is associated with mood, and it makes us feel calm and content and satisfied. So normally serotonin is taken back up by the presynaptic neuron, and then serotonin reuptake inhibitor drugs that treat depression prevent that reuptake. Well, acetylcholine doesn't get taken back up by the presynaptic neuron. It is broken down. So acetylcholine has a different process. Acetylcholine will be broken down by an enzyme called acetylcholinesterase. Enzymes always end with ASE. So it will be broken down. So then when that neuron needs to release more acetylcholine because there was another action potential, then that neuron needs to make more acetylcholine. Now that we've had our action potential in the muscle cell, the action potential travels through the T-tubule. Now we're going to shift our focus and we're going to look at what is happening with those muscle filaments. Now we're zooming in even more. We are looking at the myofibril, which is bundles of filaments. These myofibrils are composed of thick filaments and thin filaments. We're going to zoom into what those look like. And you can kind of see here that here's a thin filament, and it's connected by this Z disc, or sometimes called a Z line. And there are thin filaments branching off in both directions. And then there are thick filaments here that are connected by this M line. And these thick and thin filaments overlap each other. So now we are looking at the thin filament, and this is the thick filament. So when we look at how the thick and the thin filaments overlap, they're composed of different proteins. So let's start with the thick filaments. The thick filaments kind of look like if you could take two golf clubs and twist them around each other and have the two heads sticking up. That's kind of what myosin looks like. Okay, so the myosin is the main protein that is composing the thick filaments. And the thin filaments and the thick filaments slide across each other. And when they slide towards the center of this individual unit called a sarcomere, then that makes the whole muscle shorten, which is contraction. Now, the thin filaments, they are composed of 3 different proteins. The main contractile protein we call actin. So the myosin is going to bind to the actin and make it slide towards the center of the sarcomere. So actin and myosin are the contractile proteins. But there are 2 other proteins, and we call those regulatory proteins. And they are troponin and tropomyosin. So when we zoom in a little bit more, we're going to look at how all of those proteins interact with each other. So one single sarcomere is from one Z disc to the other Z disc. This is one sarcomere. And sarcomeres are connected to each other all along the whole entire myofibril, through all the myofibrils in every cell of every fascicle of every muscle. So now if we put a bunch of sarcomeres together, here's a Z line, here's a Z line. So this is a sarcomere, and this is a sarcomere, right? So we have a whole bunch of sarcomeres put together. Wherever there's thick filament, that's the A band. Wherever there's only thin filament, that's the I band. Wherever there's no overlapping between thick and thin filaments, that's the H zone. The M line is the center protein holding those together. If we look at the components of the thick and thin filaments, the thick filament is only myosin, and the thin filament is 3 proteins. So the main contractile protein is actin, and that looks like a sort of beaded necklace that's twisted. So all these spheres are the actin monomers, and this is what the myosin heads are going to bind to. Now, the regulatory proteins are called troponin and tropomyosin. These don't themselves contract, but they play a role in regulating contraction. The actin has myosin binding sites, these very specific spots on the actin where the myosin head is going to bind. Now, the tropomyosin covers that up when muscles are not contracting. So when the tropomyosin is blocking these myosin binding sites, the muscle cannot contract. So how do we move that tropomyosin so that the myosin binding sites are available? Okay, now the troponin is going to come into it. Troponin binds to calcium. So when we have the action potential in the muscle cell, the action potential goes through the T tubules and it lies next to the sarcoplasmic reticulum. What's in the sarcoplasmic reticulum? Calcium, lots of calcium. So the action potential triggers the release of calcium from the sarcoplasmic reticulum. Now the calcium can bind to the troponin. When it binds to the troponin, it makes the tropomyosin move. Then the binding sites are available. Then the myosin can bind. So without the action potential, there's no calcium. Without the calcium, the myosin can't bind to actin and you can't have contraction. And that works out well because then we only contract muscles when we want to. Now let's look at what is happening to the sarcomere during contraction. So here we have a relaxed muscle, and you can see that we have these I bands, and we have the A bands, and they alternate, A, I, A, I. The zone in between here, where there is no overlapping, that's our H zone. Now, what regions are going to get shorter? So when we have contraction, the thin filaments are always moving in towards this M line. So in this sarcomere, this thin filament is moving that way, and this one is moving that way. And it is going to carry on all throughout all the sarcomeres, and eventually they will all start to come closer together. So what changes? The H zone is going to get smaller because the thin filaments are moving in. And the I band is going to get shorter because as these move towards the M lines, the myosin and the actin are going to be overlapping more. And so then it's going to look like this. We can see that as the thin filaments move across the thick filaments towards the M line, the Z lines are getting closer and closer together. Each sarcomere is getting shorter. Notice that the A band does not change. This is the length of the myosin proteins, and that doesn't change. So what changes is the amount of area of the sarcomere that is overlapping. So only the H zone and the I band will get shorter. The A band stays the same. So now that we've looked at how the actin and the myosin slide across each other, I want to focus just a little bit more detailed on how the troponin and the tropomyosin play an important role in allowing the contraction to occur. So our actin molecules are the green beads. And on those actin molecules underneath here, there are myosin binding sites. Myosin, the head of the myosin proteins, can only cause contraction when it can bind to those binding sites. But in a relaxed situation, when the muscle is not contracting, there is no action potential. So there is no excitation. So the binding sites have to be blocked, and they are blocked by tropomyosin. The only way to move the tropomyosin off of these binding sites is by having calcium bind to the troponin. When calcium binds to the troponin, it causes a conformational change in that protein that pulls the tropomyosin off of these binding sites. Now, when the binding sites are available, then the myosin head can attach. And then when the muscle is contracting and has the power stroke, then the myosin head is going to move and it's going to push this actin filament towards the M line. Now, we have one more little bit of detail, and that is energy. We can't contract muscles without ATP. So how does ATP play a role in muscle contraction? And that is all about the myosin head. So remember that when we use an ATP molecule, adenosine triphosphate, there's 3 phosphates. When we use ATP for energy, the last phosphate breaks off. And when the phosphate breaks off, and when it binds to another protein, when it unbinds to that protein, all of those little changes cause proteins to change their conformational shape. And that's exactly what's going to happen with the myosin. ATP is going to bind, the phosphate is going to come off, and that process is called hydrolysis. It's going to break that phosphate molecule off. When the phosphate breaks off, it's going to make the myosin change shape, and when the myosin changes shape, it's going to move the actin. So there's very specific steps, and this is called the crossbridge cycle. When the myosin binds to the actin on the myosin binding sites, that is forming a crossbridge. And when the myosin moves, that is the power stroke. So we're going to look at how that happens with ATP. Here we have a myosin head. So our thick filaments are composed of lots of myosin molecules, our sort of double-headed golf club shape here. And these myosin heads need to be able to bind to the myosin binding sites on this actin. This is a resting position. We're not contracting at this point because there's no connection between the myosin and the actin. But notice that there is an ATP molecule that is bound to the myosin in the resting or relaxed state. Now, that ATP is going to hydrolyze. When it hydrolyzes, it becomes adenosine diphosphate and a phosphate molecule, and they stay stuck to the myosin head. Now, in order for contraction to occur, we have to have our action potential in the T-tubules that cause the release of calcium from the sarcoplasmic reticulum. That calcium is going to bind to the troponin, and it's going to move the tropomyosin off of the binding sites. Now notice the myosin in this stage here, this shape that it's in. When the ATP hydrolyzes, it causes the myosin to move back into this sort of ready position. It's still not bound, but now we have calcium. The binding sites are going to be available. Then the myosin is going to bind to the myosin binding sites, and that forms the crossbridge. When the phosphate leaves, this triggers the power stroke, and this is going to cause the myosin to move back into its normal original conformation, and that causes the actin to slide towards the M line. Once the power stroke occurs, then the ADP is going to come off. And at the end of the contraction, notice that the myosin and the actin are still bound together. They cannot unbind until a new ATP molecule binds to the myosin. When the new ATP binds, then the myosin can be released from the actin. And this cycle will continue as long as there is an action potential stimulating the release of calcium. So ATP is required for the myosin to be removed from the actin so the muscle can relax. Now, ATP is actually required for two reasons. Okay, so our muscles use a lot of energy. Okay, partly because every single time myosin binds to actin and moves it, every single little myosin head movement requires an ATP molecule. This is why your muscles use so much energy. But there's a second place where ATP is required. So all the calcium that was released from the sarcoplasmic reticulum because of the action potential, that calcium can't just stay there. Otherwise, that myosin, every time a new ATP binds, it's going to want to stick and it's going to want to contract again. So when muscles need to relax, we have to also get rid of the calcium. So the troponin can move the tropomyosin back over those binding sites. So how do we move the calcium back into the sarcoplasmic reticulum? We use active transport. Active transport uses ATP, so we need to have energy to move the calcium out of the cytoplasm and back into the sarcoplasmic reticulum. When we're going to contract a muscle, let's suppose we're going to contract the bicep. If I pick up a pen, how heavy is that compared to if I pick up a dumbbell? The dumbbell is heavier than the pencil. How do I control both movements? It's based on the number of muscle fibers that are recruited. So when the load is heavier, then more muscle fibers need to contract. So we talked about how a motor unit is when one motor neuron connects to multiple muscle fibers. When I lift the weight compared to the pencil, I need to have more motor units recruited. So that's about the nervous system. So then the frontal lobe, your motor cortex, has to signal more motor neurons so that I can lift the heavier weight with more muscle fibers being engaged. And when we have a sustained muscle contraction, so let's suppose I'm going to just hold the weight for a period of time. As long as there is nervous system stimulation, the muscle fibers are going to contract. And then the myosin is going to keep binding. It's going to bind, new ATP will attach, it will bind and contract over and over again, kind of like looking at a millipede, how it walks. All of those myosin heads are going to continue to bind and release as long as there is an action potential stimulus keeping the calcium present. And the last thing that I want to just point out is there's different kinds of movements. So we have a concentric contraction, and that is when, if we look at the biceps, I'm going to contract and flex my arm and contract the biceps muscle. But there's also eccentric, which is when I lower it. So now, what's actually happening with the actin and the myosin in that case? Actin and myosin still have to bind, there's still ATP, there's still an action potential, there's still calcium. But now, every time the myosin binds and releases, the sarcomere is getting stretched out, right? So in a concentric movement, the sarcomeres are getting shorter. In an eccentric contraction, The sarcomeres are getting longer, but that whole contraction cycle is still occurring. And then if you hold the muscle, so my muscle is contracted, but it's not moving in either way, it's isometric. So the sarcomeres only technically shorten during a concentric contraction. So I made a summary chart for you that shows the list of steps of everything that is occurring from an action potential in a motor neuron to the contraction of the actual muscle filaments inside the myofibrils of the cell of the muscle.